This is going to be the last set of videos for microevolution, which means that after these videos you will be having a test on microevolution. I want to rewind just to make sure that we are familiar with the Hardy-Weinberg equilibrium and the equation that goes along with it. We've been doing a lot of practice problems with this, and so what's important is that you know, A, the 5 conditions of the Hardy-Weinberg equilibrium, which is the situation in which we would be able to use the Hardy-Weinberg equation. So remember that equilibrium are the 5 conditions, and the equations are the actual 2 equations that we've been working with. So we can talk about like the Hardy-Weinberg model when we talk about frequencies. And so this overlaps the Punnett square and the Hardy-Weinberg model. Essentially what happens is once we find P and Q, which is going to be the dominant allele frequency and the recessive allele frequency, what we do is we set it up on a Punnett square, but we also use those values for P and Q. And so we can see in the top left box, CG, or we'll say that that was P, was 0.6, and P was also 0.6. On the other side, when we multiply those two together, the frequency of having a dominant homozygous individual is 0.36 out of all of my offspring. If you'd like me to go over this more, we can talk about this more, but what's important is that this adds the numbers to the Punnett square. This is how they derived it. We do not need to know how to derive the Hardy-Weinberg equilibrium equations. Moving on to other information that we need to know about microevolution. The idea of natural selection is that it is the only mechanism that consistently causes adaptive evolution. Evolution by natural selection is a blend of chance and sorting. Basically what this means is that chance means that I am having some sort of mutations that create new genetic variation, and sorting means that selection is always going to favor some alleles over others, which means like I am more fit than the other organism. This means that natural selection is in part due to the environment and in part due to our genetics and our chromosomes and our mutations. When we talk about the sorting part, or the alleles that are more favorable, we want to talk about relative fitness. There are animals and species, there are organisms within a species that are always competing. Most likely those are males, and reproductive success is what dictates how fit I am for my environment. Relative fitness is defined as the contribution an individual makes to the gene pool, and so truly the only way that an organism contributes or is considered to be fit is if they can reproduce and create offspring that are also going to survive to reproduce. If they are not, it means that they are not strong enough or they are not fit enough within that environment, and their genetics should not be passed on. This is basically how we get rid of certain genes eventually. There are 3 modes of natural selection within a population. Natural selection alters the distribution of the alleles, basically what the organism looks like. The 3 are directional, disruptive, and stabilizing. In directional selection, it basically means that, right, I have a phenotype, fur color, and I know that there are certain genotypes that result in that. There's probably a dominant genotype for brown, or like dark brown, a recessive genotype for white. Usually, most individuals lie somewhere in the middle of that color scheme. As we see on the left, it's a bell curve, which means almost all the individuals are the light brown, where there are very few that are really, really light or really, really dark. In directional selection, we see that for whatever reason, the average starts shifting to one side. And so we see that in this case, the average is shifting towards the darker side. There are still only a couple that are truly very dark colored mice and truly very light colored mice, but the average has shifted more towards the darker side. The possible effects of continual directional selection is we start losing genetic variation. We see that on the far right, not only is the bell curve moved over, it's also very narrow, which means that I have fewer individuals in those areas, which can be a dangerous thing. Disruptive selection is going to occur when there are conditions that favor the 2 ends of the bell curve. So the really dark and the really light mice, and the intermediates are going to start to die off and not be able to reproduce. And we see that there's basically a big dip in the center of my bell curve at this point. The last type is stabilizing selection, which basically means that any organisms on the far ends of the bell curve start to die off and don't reproduce. This type of selection reduces variation because I'm losing the really dark and the really light phenotypic characteristics, and most of my organisms are kind of hitting right in the middle. At the time, this is not terrible, but when I lose those genetics and I can't get them back into the pool, if for some reason my mice need to be darker colored to maybe avoid predation, those genetics are not available anymore. To talk a little bit more about sexual selection, we've talked about this already, so this should be review. A form of selection occurs when individuals are more likely to choose certain individuals as mates because of certain phenotypic qualities. This can result in sexual dimorphism, which is the physical differences between the two sexes. Obviously, human males and females look very different. When we look at deer, some have antlers, females do not. Sexual dimorphism is very common in birds. You can see the quails in the bottom left. This does not always occur, though, only in some species. How does this sexual selection operate? Intrasexual selection occurs within the same sex. So that means that males are competing with each other. That basically means that the organisms of one sex are competing for mates of the opposite sex. This is pretty much always males all the time. Intersexual selection, mate choice, is when individuals of one sex are basically choosy about who they mate with. This is usually the females. The males compete with each other, and then the females can either opt to mate with certain individuals or not, based on their showiness or their strength or their size and things like that. And so typically, not always, males show intrasexual selection, females show intersexual selection. How do we preserve this genetic variation? Because we've talked a lot about how if we lose this genetic variation, that can be a really difficult thing for a population. Some of the genetic variation in populations represents neutral variation, differences in DNA that don't provide a selective advantage or disadvantage. There are several mechanisms that counter the tendency for directional or stabilizing selection, which end up kind of getting rid of the extremes or the variation. The four things that are listed we are going to talk about: heterozygote advantage, frequency-dependent selection, balancing selection, diploidy. All of these things to help us preserve genetic variation, which is important for the survival. Diploidy is, in diploid eukaryotes, each organism has 2 copies of every gene. So we have sets of genes, right? For gene 1, I have 2, one from my mother and one from my father. A considerable amount of this genetic variation is hidden in recessive alleles, and so there is a lot of variation, but it is not always shown phenotypically, but it may be passed on. Often those alleles are recessive and less favorable, but they can be hidden. They can still be passed on, but they can be hidden, which means that it still maintains variation. A haploid organism only has 1 chromosome for every gene, and so everything on that gene is expressed. Diploidy basically means that recessive alleles can be propagated or passed on through heterozygous individuals. I can have a dominant allele that covers up a negative allele, and if I'm heterozygous, no one will ever know until I potentially pass that on. We call this latent variation or unseen variation, and it is only shown if I mate or an individual that is heterozygous also mates with another heterozygous individual, and by random chance, both the egg and the sperm contain the recessive allele, and then the organism's offspring would get the 2 recessive alleles. As you might expect, this happens very infrequently, which leads us to talk about balancing selection. Balancing selection occurs when natural selection maintains 2 or more forms in a population. Basically, this type of selection includes heterozygote advantage and frequency-dependent selection. Heterozygote advantage means that an individual who is heterozygous for a gene is more fit than the homozygous individual, even if the gene is not negative, right? Because a homozygous dominant individual could have both of the positive genes, but we find that often the heterozygotes who have more variety in their gene pool are more fit, therefore passing on more genes. This is basically true of any organism that has a recessive allele that is dangerous. For example, sickle cell anemia is caused by the inheritance of two recessive genes from both parents, and it makes the blood cells form a sickle cell, which is like a curved moon-shaped cell. For these homozygous recessive individuals, they are very sick, shorter life expectancy, etc. For someone who inherits one recessive trait and one dominant trait, so they're heterozygote, they are considered carriers. They may suffer problems from time to time, but they do not experience the actual disorder. Interestingly enough, someone who is a heterozygote for sickle cell anemia is immune to malaria, which is a parasite that usually is present in Africa. For whatever reason, these heterozygotes are more fit when it comes to malaria than someone without the disease completely or with the disease, sickle cell anemia. And so we see here where we see the frequencies of the heterozygote advantage versus disadvantage. The idea of frequency-dependent selection indicates that the fitness of a phenotype depends on how common it is in the population. The positive frequency-dependent selection, or in this positive frequency-dependent selection, the fitness of a phenotype increases as it becomes more common. The negative frequency-dependent selection, in negative frequency-dependent selection, the fitness of a phenotype increases as it becomes less common. So there are two scenarios that we are talking about, and this is totally dependent on the type of population we are looking at. Either a very common genotype makes an organism more fit, or having the less common phenotype makes them more fit. An example, prey switching, rare morphs of prey are actually fitter due to predators concentrating on more frequent morphs. And so we see a negative frequency-dependent selection occurring here. If I look very different than all of the other organisms, the prey are not going to see, or the predator's not going to see me, they're going to prey upon all of my counterparts. Another way that we maintain variation, balanced polymorphism. This occurs in a given population when two distinct types exist and the allele frequencies don't change. This is due to the fact that variation in the environment might favor both morphs. We call the phenotypes morphs, or one morph may be more adapted to survive during a certain time of year over the other. And so we see that some, like the ladybugs, the black ladybug will be very prevalent during the spring. The red ladybug will be very prevalent during the fall, and they switch on and off all year. Why natural selection can't eventually fashion perfect organisms. So we see these selection mechanisms occurring all the time. Why do we not eventually end up with these perfect organisms? Selection can only act on existing variations, and we know that mutation is always going to create new variations, new advantageous alleles. Evolution is also limited to historical constraints. So evolution does not scrap the ancestral anatomy. Basically, they're already starting from what they have. They can't totally get rid of traits. Also, adaptations are often compromises. A loud call from a frog attracts mates, also attracts predators, so being too loud is also a disadvantage. Also, chance, natural selection, and the environment interact. We know our environment is ever-changing, and this can be a really big issue when we're trying to fashion the perfect organism. That is the end of the PowerPoints for microevolution. You need to study this material for your test.